Within the muscle, you have the subdivisions, the fascicles, which have the muscle fibers inside them. What you're going to look at today on figure 11.2, those are the final structures within each muscle fiber, meaning the muscle cell. So what are the further divisions of the muscle fiber? What do we have? If you did look up, I did ask you to look up while you were waiting.
Response.
What are the subdivisions of the muscle fiber? The next level.
Sarcopenia, Fauci.
Before that, you have to first be able to study the picture. Okay, what you are looking at?
The endomysium.
That's connective tissue, that's way off. I'm asking you, the muscle is arranged into muscle fibers in groups, fascicles. What you are looking at is a single muscle fiber, and you have further subdivisions within that. So if you looked up the description and the figure, you would understand. Okay, without wasting further time, myofibrils and myofilaments, those are the further subdivision. I didn't ask for the different parts in the cell. Okay, so the myofilaments is the next level, and within that you have myofibrils. Can you at least see them? Can you at least locate them? If you are on the right page. Okay, thank you. Then you have other parts of the cell, like any other cell. If you forgot the cell parts, go back and review them when you get a chance. Okay, so like any other cell, you see the nucleus, mitochondria, and all of that, but only some of the names they call it a little different. Whenever you see the word myo, that refers to the muscle, and when they consider the muscle cell more at that level, they use the word sarco. So cells have the plasma membrane, they call plasma lemma. Here, what do we call that membrane? Sarcolemma. All right. So what do we call the cytoplasm of this cell?
Sarcoplasm.
Sarcoplasm. What do we call the ER, the endoplasmic reticulum? Sarcoplasmic reticulum. You see those blue mesh-like structure that runs parallel to the fiber. And then what are the other structures you see? You see a set of three tube-like structures going perpendicular in the transverse orientation to the fiber. So they call these 404. What do we call these structures? Transverse something. Transverse tubule and transverse cisternae. So you have in the middle, you have the T-tubule, the transverse tubule. On either side, you have the transverse cisternae. Okay. Then you see those dark and light shaded areas, the purple colored, and you see the orange colored areas, those are the gaps. And then you see a wavy line in the middle that's supposed to be your Z disc. So they have the A band, I band, and Z band. A band covers the purple lines which represent your myosin. The I band is between the adjacent myosins, and the Z disc is where you see that wavy line. But we will see it more clearly in the following pages. Okay. So before we talk about the myofilaments, I want you to go to the previous page and look up some of the terminology. We talked about the sarcolemma, sarcoplasm, myofibrils. But we have to also understand that we have glycogen and myoglobin in these cells. Glycogen is a polysaccharide that's a stored form of glucose, like in the liver. You find it in the muscle cells, so when the muscle needs more glucose, it breaks it down and uses it. And also myoglobin, you can relate to the hemoglobin. What does the hemoglobin do? Transport oxygen, right?
Right.
The same way myoglobin, because myo relates to the muscle, so they call it myoglobin. Then you see the terms myoblasts and satellite cells. What are myoblasts? Like remember your osteoblasts, they come from the stem cells. Here the stem cells that make the muscle cells, they call them myoblasts. And then sometimes they are in the transition stage before they become fully developed into the muscle cell. They call them satellite cells. They're ready to transition when needed. Okay. And then you see all the other structures highlighted that we discussed. I'll give you 2 minutes to make sure you go over these names and locate them if you didn't do it when you had the chance. Then we will talk about the myofilaments. The myofilaments are within the myofibrils. So as you can see in the next page, you have thin and thick filament. Thin and thick filament because of the size. Okay, the thin filament is only 2 strands. The thick filament has multiple strands. But if you take the individual units, you will have 2 strands with 2 heads, 2 bulb-like structures. It looks like a golf club. Okay, but you have several of them, hundreds of them together. That's why they call it thick filament. But we need to get used to these words. The thin filament is your actin. Okay, that's your thin filament. The thick filament with several strands and the heads, they call it the myosin. That's your thick filament. And what else do we have on the thick filament? You have the myosin head. What about the thin filament? What do you see? Do you see some spaces? It's arranged like beads on a chain, individual beads. The whole thing they call fibrous or F-actin. And the individual bead-like structures, you need to use the book because this what is much more clear than what I have. This is just for me to help you walk through the chapter, but you have better pictures in your book. Okay, so if you see those individual beads, you see some cavities that are covered actually by that bluish structure with the yellow structure on top. So those are your active sites. These active sites are the spaces where the myosin heads will bind in order to move your muscle at the microscopic level. Okay. So there are 2 more proteins that are involved. The tropomyosin that goes around your actin, and the yellow knob-like structure, that's another protein called troponin. The 2 proteins that are involved with muscle contraction, we call them contractile proteins, and the ones that are involved in regulating the muscle movement, when to move, how to move, that's called the regulatory protein, the tropomyosin and troponin. So I want you to take a couple of minutes and make sure you understand these different myofilaments. What is actin? How does it look like? What is myosin? What's the difference? And what are the other 2 proteins that you find attached to the actin? And the actin has 2 names, F-actin and G-actin. Which one is what? And then in the last part of the same figure, you can see the alternate arrangement of the thin and thick filaments with the empty or bare zone in the middle. That's what we are trying to close. This is at the microscopic level. But we will learn some more specific names in a couple of minutes. If you turn over the page, you will see in the next figure there are some more proteins that transmit the movement all the way to the endomysium on the outside. And of these other proteins, protein of clinical relevance is called dystrophin. You may have heard of striations when you try to look at the three types of muscles, the smooth muscles, skeletal, and cardiac. Which type of muscles have the striations?
The cardiac
And skeletal, right? The striations come from this alternate arrangement of the thin and thick filament. They have a pattern, that's why you see the striations. In smooth muscle, you don't see it because it doesn't follow a certain pattern. So if you see here in figure 11.5, you can see how the actins and myosins are arranged alternately. And you can see a thin green spring-like structure that attaches. It extends from the Z disc to the actin. Okay, it basically anchors the actin on both ends to the Z disc. So this whole set between the adjacent Z disc we call sarcomere. But you have multiple sarcomeres above each other and on either side. There could be several units of sarcomeres. So each sarcomere is considered the basic structural and functional unit for muscle tissue, like the osteon for the bones, if you still recall. Okay. Then we need to understand how they call these different regions as A band, I band, H band, M line, Z disc, and all of that. So where is your A band? A band covers the distance of the actin, the length that is covered by the actin. If you look up, the arrows that covers the A band, right? And then between the adjacent actins, not the actin, the myosin. The purple color structure is the myosin, that's your A band. But between adjacent myosins, you have that gap, that's your I band. Then within the A band, there's a gap which we saw in the earlier figure as bare zone. Here we have the gap between the two actins, adjacent actins, that's your H band. The H band is where we are going to actually do the contraction within a single sarcomere, but it happens in multiple sarcomeres at the same time, so your muscle contracts. And then you can see the M line, that's right in the middle of the H band. The Z discs are obvious. Okay. The green filament you see that attaches from the Z disc to the myosin, that's called elastic filament. It's made of a different protein called titin, T-I-T-I-N. So you should know the different regions, what's the span for each region, and what do we call the sarcomere. Sarcomere extends from where to where. So it's basically the segments between two Z discs is your sarcomere. Can you see that? Okay. Then moving on to the next section, we have to talk about motor units. What are motor units? If you took the lab already, you know that. What is a motor unit? Can anybody help me with that if you have read?
It's a neuron.
You're halfway there. Neuron plus. You can still look at the picture, the figure there until you don't have to read, but you can see what can you guess. It's a single neuron and all the muscle fibers activated by that neuron. It's called a motor unit. Okay, so you have the somatic neurons that come and activate your skeletal muscle fibers. So you can see in this figure 11.6, you can see two nerve fibers, and the neurons, motor neuron 1 and motor neuron 2, you see they are going to different muscle fibers, right? Usually each terminal of a single neuron ends in a single muscle fiber. One neuron can put multiple, like thousands of terminals and connect to thousands of fibers, or multiple neurons can go to the same fiber or put fewer terminals to fewer fibers. It depends on where and what kind of action is involved. Those details, we'll get that later. Okay. Okay, so what I'm trying to say is that the way one neuron connects to the individual muscle fibers can vary depending on the location, on the function, on the composition of the muscle fibers. There are fast, slow, intermediate muscle fibers depending on the organization. That's towards the end of the chapter. So when a neuron makes a contact with another neuron or with a muscle fiber, in this case a muscle fiber, the contact is not physical. Why? What kind of signal does a neuron carry? Throughout the main part of the cell. Chemical is at the end of the neuron.
Oh, yeah.
Electric signal. So basically you have electric signal, towards the end you have chemical signal, release of the neurotransmitters picked up by some receptors on the other neuron, or in this case the muscle, okay, and then some action happens, okay. Then again you will have electric signals passed down the muscle cell or another neuron. So chemical, electric, you have alternate signals being passed on. So this leads us to the neuromuscular junction. So when you say neuromuscular or myoneural junction, the same thing, they use the terminology both ways. So I want you to take a couple of minutes and try to understand what are the structures involved. You can use figure 11.7. Then we will discuss about that. You see one end of the neuron coming in contact with the muscle fiber cell. On the side of the neuron, you have the very tip. You call that structure synaptic knob. Synapse refers to the junction between two neurons or a neuron and a muscle, the junction. There's no physical contact. There's a little tiny gap. Okay. So you can see the basal lamina surrounding and separating the synaptic knob from the rest of the surrounding tissue. And you can see the Schwann cell and all of that. And then you see these green round dots that represent the vesicles that contain the neurotransmitters. So the neurotransmitter chemicals will be released once you have the electric signals coming down all the way to the tip of the neuron, and then these vesicles will merge with the membrane and release the content in the synaptic cleft. Synaptic cleft is the actual gap. Synapse is a generic term, synaptic cleft is more specific for that gap. Okay. So when these vesicles release the content, the neurotransmitters, it could be acetylcholine represented by ACH. The abbreviation ACH stands for acetylcholine, and there are other neurotransmitters we will learn when we do the nervous system. So in this case, we are discussing the acetylcholine. So when these vesicles release the acetylcholine, what process is happening? Go back, rewind.
A contraction through your muscles.
Rewind to chapter 1 or 2 or 3. I asked you what
diffusion.
process the transport. Can you be more specific?
Like osmosis or diffusion.
What are we looking at? We are looking at vesicles. What do vesicles do? Thank you. Exocytosis. It's exiting. It's releasing the content to the outside. Is it passive or active?
Active.
Active transport. Okay, whenever you use vesicles or whenever you use energy, you consider it as active transport. Diffusion, osmosis are all basic transport processes, but you have to relate information. See, we learned that for a reason, because it helps you here. If you forgot, that's a different thing. Okay, you can always go back and read up. Okay. So once the chemical is released in the synaptic cleft, what do we see on the other end? I don't know if you see the name, but the other end where you see those structures on the muscle fiber, they call it motor end plate. And here you see those junctions. You see those foldings? Those are called the junctional folds because they are found at that junction. And what do we have in those junctional folds?
Receptors.
Yeah, thank you for reading. ACH receptors. So you need to have somebody to receive him, right? So the acetylcholine is received by the muscle fiber through the ACH receptors. Okay, so far so good. Okay, that's what you see on the slide as well in the inset. Okay. All right. And then if you turn over the page towards the end of that paragraph, you also see an enzyme called acetylcholine esterase, ACHE, that destroys the acetylcholine. Because once the acetylcholine is released in the synaptic cleft, it binds to the receptors, it's going to start something downstream, okay, action. So if you keep doing that, your muscle will be stuck. It will remain contracted. So how do you release, relax? So you need to remove the acetylcholine. Somebody has to do that. So the acetylcholine esterase is the enzyme. Once the chemical is released, within a short time it's removed as well. But again, you will get more repeated signals from the nervous system if you need to do something more and more, how long you need to do. Okay, the next aspect is electrically excitable cells. Under that section, they discuss the electrophysiology. The electrophysiology deals with electrical conductivity of the cells. How do we generate electricity? What do we need? Again, go back, rewind.
Heat.
Electricity.
Oh, no.
Don't say you know. Don't say I don't know. Think.
Muscle fibers?
What do we have in a battery? How do you generate electricity? You have two things. Yeah, charge. So what do we have? What can we have in the cells that have charge? Ions. Can you be a little more specific? Electricity, ions. Can you link them with one word? Yeah, that's good. Electrolytes. Thank you.
Yeah.
Electrolytes. Electrolytes conduct electricity. Okay, these are compounds that dissociate and produce charged ions, plus and minus. Depending on which side is more negative, positive, you have certain level of electric current in our body itself, right? So inside the neurons, inside the muscles, everything is done by electric signals. Okay, by the way. So your muscle cells normally remain negative inside. When necessary, when the muscles have to contract, it becomes more positive inside and then goes back. This is done by shuttling of the ions in and out. We'll see which ions in a couple of minutes. First of all, that's what they discuss under electrophysiology. Polarized, meaning one side being more negative versus the other. The inside is usually polarized. And then resting membrane potential. That's the electric current that you will have normally when your muscles are at rest. Then you see depolarization, action potential, repolarization. So depolarization is when you reverse this negative charge to more positive charges, okay, inside of the muscle cell. That's when you are going to have the muscle contraction, okay. And then you will have action potential. So the electric potential is going to be more positive, let's say from minus 70 to it becomes more positive values like plus 35, plus 55 and so on. So that's when you have action potential. It's nothing but the electric potential you need for muscle to act, meaning to contract. Okay. And repolarization is once the contraction is finished, it goes back to the original state. That's when they call it repolarization. These three terms can be confusing. Once you get it correct, it should not be a problem. Polarization, depolarization, and repolarization. If you know one very well, meaning polarization means inside the muscle cell is more negative. Depolarization is the reversing of that. and repolarization going back to that. And resting membrane potential, nothing is happening. Action potential when your muscles contract. Okay. Let's take 5 minutes. I want you to look up pages 413 to 417, hoping that you know all the basic structures involved here. If not, you can review that and look this up. I want you to go through step by step, try to understand, and then we will discuss it together. How the muscle contracts and relaxes. Please look up the pictures for now. If you read the description, as long as we can do quick, otherwise it will take forever. That's something you can do at home. You should be able to understand with just the pictures if you know the parts. Just go through the steps so we can do it together. Otherwise, it will take a while. I'm just waiting for you to look through all the steps, not read through the content. That will take more time. That's where reading before class helps. All right, Brunel, what's happening in step 1?
Just showing that
Go with the pictures, okay? Read whatever is in the pictures.
arrival of the nerve signal.
That's what you need. First of all, your nerve has to activate everything. So meaning the process starts with arrival of the nerve signal at the terminal end, which is the synaptic knob, right?
Right.
And then calcium rushes in, and the calcium leads to the next step, which is the fusing of the vesicles with the membrane and release of the acetylcholine, where in the synaptic cleft. So what's the next one?
Oh, me? Where it's binding?
Just please read the step.
Okay. The step 2 is accelerate. There, I can't read that word.
Acetylcholine.
Acetylcholine release.
Okay, abbreviated as ACH, but we need to get used to the word acetylcholine. So what's happening in step 3?
Binding of the ACH to the receptors.
Binding of the acetylcholine to the receptor protein on the other side, on the muscle cell fiber. This induces the protein to open the gates. Remember the gated proteins, gated channels that allow different electrolytes in and out when we discussed the active transport at the beginning. So here, what are the other two elements that are involved, the electrolytes or the ions? Sodium and potassium. So which way is sodium going in? I mean, I said that already. Okay. So which way is potassium moving? Out. Okay. So this is a protein that transports two substances at the same time. And you remember the symport, antiport, and all of that. Antiport meaning two substances moving in opposite directions. So everything connects, okay, whatever we learn. So if you see that happening, this shifts the charges, because every time 3 sodium goes in, 2 potassium is pumped out. So you're putting more and more and more sodium in. So in the resting membrane potential, what did we have? The charge, overall charge inside the muscle cell, was it negative or positive?
Negative.
Negative, right? So when more and more positive sodium, positively charged sodium comes in, it's going to become more and more positive. So let's say initially you had a minus 70 or a minus 90 millivolts. So when more and more positive charge is coming in, this is going to go up the electric potential to say about plus 35. It can go as high as plus 55 or more. So this is the minimum value you need for an action potential to occur. They are going to call this the threshold value or the threshold potential, and between this and this you have the action potential. Okay, so now it was originally polarized, now it's getting depolarized as the sodium comes in, and then the sodium stops, and then when the muscle contraction is finished, potassium is going to leave, and then it will repolarize again because more positive charges are now leaving. Okay, so if you need to know more, you can read up on the sodium potassium pump. You have it in the book, or I have it in my website, how the pump works. You can go and look up that video. Next step, Bruno.
Says right here, the opening of the ligand regulates ion gate,
Right.
creating the end of plate of the potential.
Yeah, that's what we just discussed.
Right.
Next.
Okay. Open.
6.
Oh,
Step
the
6.
6. The action potential is propagate down the
Come
tube.
on, you should at least help me here so I can help you. Okay, action potential is being transmitted through the transverse tubule. You see the yellow arrows going down?
Yes.
So the T tubule that's in the middle, so that's transmitting this action potential across the muscle fiber. And we have calcium stored inside the sarcoplasmic reticulum, by the way. So that calcium, see the yellow dots, now they get out of they diffuse into the the cytoplasm of the cell. You see the yellow dots coming out, right? In the next step. So once the calcium diffuses, it randomly binds to your troponin. You know where is your troponin? On the tropomyosin, on the actin, on the outside. If you remember the previous figure, you have it here also in step 8, right? So once calcium binds to the troponin, it's like an on-off switch. When calcium binds, it's like you're triggering something, you're turning on something, okay? So what's happening in the next step?
So the shifting of the troponin exposure active.
Tropomyosin. You can read slowly. The first time you learn, you will get it.
Right.
Get tropomyosin.
Tropomyosin exposure of the active sites on
Exactly.
actin.
So the shifting of the tropomyosin, it moves a little bit. So what are we exposing? You see those little cavities in those beads, the G-actin globes. You see the gaps. Those spaces are the active sites. We need the myosin head to go and grab right there. Okay, it has to attach there, then only it can push the sarcomere, meaning the actin within the sarcomere. So you have shifting of the tropomyosin and exposure of the active sites. So what is happening in step 10?
Step 10, the hydrolysis of the ATP and ADP plus the
Phosphate
activates and coupling of the myosin head.
Okay. So ATP is used. We need energy for this process. ATP is hydrolyzed to ADP and phosphate, and you release energy. That energy is needed for the cocking. You see the position of the head, it was sleeping, kind of sleeping. You don't use that word, but it was kind of resting. Okay, now it's cocking, C-O-C-K-I-N-G. So it goes up and then it attaches to the active site on the actin. And it makes a structure, transient structure, we call it cross bridge. You can see the word in the next step. You see the cross bridge?
Yeah.
Yeah, the cross bridge formation occurs, and then it does a power stroke. We are looking at one myosin head, but you have so many, right? So when this happens across your muscle fiber, your sarcomere first contracts, the actin slide over the myosin. So they also call it sliding filament theory because the actin slides over the myosin back and forth. So when the power stroke occurs, it slides over. So the gap is closed. All right. So you can see that at the bottom of that page. And then, if the muscle contracts and if it stays there, we need relaxation, right? It needs to come back. So how do we do that? What were the 2 steps that were critical in starting this whole process? First, what was the first thing that happened in the synapse?
The signal?
Yeah, but what did we release there?
ACH.
ACH, that has to be removed. And who removes that? I mentioned earlier there's an enzyme, acetylcholine esterase, that destroys that. Okay, so you're removing the first signal. The second signal is your calcium binding to the troponin, the on-off switch. You remember? So the calcium has to be removed. So there is a system that takes the calcium away and put it back in the sarcoplasmic reticulum. So when these 2 are turned off, meaning no more acetylcholine, no more calcium binding, then what happens? The tropomyosin slides back. You don't see the active site anymore. The myosin head cannot bind. It falls back. Okay, and your muscle relaxes. So it's as simple as that, but you need to spend some time to get all these parts figured out and all the steps understood very well. Okay. So we are going to watch a couple of videos that show this muscle action, and I have a few more videos that you can look up when you have time. And the rest of the chapter we are going to finish it up in the next class before we test the following week, Tuesday, right? Anybody doesn't know we have an exam, chapters 9 through 11? No. 10 and 11? That's more than enough, right? On Tuesday.
So I have to put down this thing right here. For sure. You know I'm going to the lab.
Right.